Hello buddies, welcome back to the channel BioPulse, and this is Chandrika. Today is day 18th of our journey, that is 40 days MCQ biology line by line NCRT, and this is the continuation part of the chapter molecular basis of inheritance. As I said, molecular basis of inheritance is a very interesting chapter and one of my favorite also. That's what I made this chapter into two portions. First part has 100 questions and second part will be having remaining 100 questions with respect to particular chapters, particular topics. Okay now, so today is the part 2 of the chapter molecular basis of inheritance. So let's begin. Here is the first question of the part 2, 101 question. Choose the correct statement. A. Monocistronic eukaryotic structural gene have interrupted coding sequence. B. Polycistronic prokaryotic structural gene have interrupted coding sequence. C. Monocistronic prokaryotic structural gene have interrupted coding sequence. And then D. Both A and B. So here the right option is option A. Monocistronic eukaryotic structural gene have interrupted coding sequence. What is monocistronic? What is polycistronic? Monocistronic means the gene which is responsible for the formation of one kind of protein. Okay now, poly in the sense what? Many proteins will be responsible by that gene. Okay now, here I have made a diagram. Here you will be having a clear cut appearance of what actually polycistronic and then monocistronic of the mRNA. Multiple genes in one mRNA is called polycistronic. One gene in one mRNA is monocistronic. Poly means multiple, mono means one gene. So you can keep in mind like this shortcut. Okay, what is polycistronic and then what is monocistronic? See, let me explain. Polycistronic mRNA means multiple genes on one mRNA. This is only one mRNA. Here this is gene 1, gene 2, gene 3. They are responsible for the production of different kind of the proteins at a time. Okay, at a time. And this kind of polycistronic mRNA you can see in prokaryotes. Usually it is seen in prokaryotes, very common in prokaryotes. Okay now, what is the key point here? One mRNA, this one mRNA has many genes. Each gene is responsible for the production of different proteins. This is protein 1, this is protein 2, like this. Okay now, if I am talking about monocistronic mRNA, it has only one gene and produce only one protein. One gene is responsible for the production of only one gene, then it is called monocistronic. Where it is common? Monocistronic is common in eukaryotes, whereas polycistronic is common in prokaryotes. So here monocistronic eukaryote structural gene have interrupted coding sequence. Yes, of course they will be having interruptions. When mRNA is formed, again it is going to be modified to form a matured mRNA. Firstly it will be having a HN mRNA. Okay, that process I can't explain here because it's a lengthy process. So here what about the B? Why it is wrong? See, polycistronic prokaryotic structural gene have interrupted coding sequence. They won't have any interruptions, so because of that this is wrong. Monocistronic prokaryotics, monocistronic won't takes place in prokaryotes, it is takes place in eukaryotes. That's what this is also wrong. So this is about first question of today's session. Just keep in mind, polycistronic means multiple gene, multiple proteins. Monocistronic means one gene, one protein. Polycistronic happens in takes place in prokaryotes, whereas monocistronic in eukaryotes. Let's move to the next question. That is next question number 102. Exons are A. coding sequence? Yes. B. non-coding sequence? No, they are non-coding sequence are introns. C. expressed sequence? Yes, when they are having the coding sequence they must be expressing. So here the option D will be correct for the 102. Exons are what actually they are expressed sequence. The region of the gene that are expressed. They appear in mature or else processed RNA, I can say. They are also called what? Coding sequence. Now next question about the introns. That is question number 103. Introns. A. Appear in mature or processed RNA? No. Exons are appeared. Do not appear in mature or processed RNA? Right. C. Appear in prokaryotes. That is also wrong. So here B is correct one. Why they are not appearing in prokaryotes? Because prokaryotes are not having any kind of the interruptions. That is, introns are absent in the prokaryotes. That's simple. Next, types of RNA and process of transcription questions. 104. Which of the following play role in protein synthesis of prokaryotes? rRNA, tRNA, mRNA, and then all of this. So here the right option is all of this. In the both eukaryote and prokaryote, if I ask, three major types of RNA are essential for the process of the synthesis. That is mRNA, which one we are calling as a messenger RNA, whereas tRNA is called as transfer, and then rRNA is called what? Ribosomal. So here the right option is option D. If I explain in detail, mRNA act as a messenger. Okay, na? It carries genetic code from the DNA to the ribosomes. Okay, and if I talk about the tRNA, it acts like an adapter molecule. It reads the codon on the mRNA. If I say this is the DNA has some information. That information is copied on the mRNA, okay, to form some kind of the protein. When DNA is giving its or else copying the information on the mRNA, this mRNA is read by whom? tRNA. Okay, na? So acts as a what? Adapter molecule. It reads the codons on the mRNA. Whatever the sequence is there on the mRNA, who is going to read by what? tRNA. And whatever the information is there on the mRNA, then who is going to synthesize the protein? The rRNA, that is ribosomal RNA, which is a major component of the RNA, which is also called as what? Protein factories of the cell. It helps in the catalyze the formation of the peptide bond between the amino acids. So next question, 105. The function of some RNA are given below. Choose the incorrect one. Oh, he is asking incorrect one. So here, which one is incorrect? A. mRNA provide template strand. Yes, of course. Messenger RNA, that is mRNA, carries the genetic code from the DNA and it acts as a template for the protein synthesis, isn't it? So next, B. mRNA provide non-template strand. This one is incorrect. And then tRNA brings amino acids. Of course it is correct, because tRNA acts as a adapter molecule. Just now I said they carry specific amino acid to the ribosome and match them to the mRNA codon. And if I talk about the D, rRNA plays structural and catalytic role. Yes, of course this one is correct, and this one is also correct. RNA plays very important role in structural and catalytic role to form the physical structure of the ribosome and acts as a ribozyme to form a peptide bonds between the amino acids. Ribozyme enzyme acts as a catalytic role, okay, and rRNA plays very important role in a structural form. How it is going to form? Because among the amino acids it is going to form a peptide bond. That's what it is responsible to play in a structural role. So here the wrong option is the incorrect statement is B. So the right option is B. 106. How many polymerases require in bacteria for transcription of all types of the RNA? How many polymerases? Polymerase is a enzyme. Okay, here the right option is A, only one. In bacteria, if I say that is in the prokaryotes, For the transcription, only one polymerase is responsible, and all types of the RNA including messenger, ribosomal and transfer. That holoenzyme consists of several subunits like alpha, beta and then sigma factors that help in the initiating the transcription specific prokaryotic sites. Okay, where if I compare to the eukaryotes, they are different from the prokaryotes because they have three different types of RNA polymerases: pol1, pol2, pol3. I will explain it later on. Okay, pol1 will be for rRNA, pol2 will be for mRNA, and then pol3 for the tRNA. But in the bacteria or else in the prokaryotes, all those things are done by only one polymerase, that is only one polymerase will be there in the transcription. Next, 107. Choose the incorrect step about the transcription. Incorrect step he is asking. A. RNA polymerase binds to promoter and initiate the transaction. So this one is correct. When transcription begins, when the enzyme RNA polymerase identifies binds to the specific DNA sequence called as promoter. Okay. B. Nucleotide triphosphate acts as a substrate and polymerization in a templates. This one is also correct, this one is correct, and then this one is correct. He is asking to incorrect step. Next, C. A short stretch of RNA remains bound to enzyme. This is wrong. During the elongation phase, what happens? As a RNA polymerase removes from the DNA, the newly synthesized RNA strand is actually displaced from the DNA template and hangs out of the enzyme. It does not remain bound to the enzyme in the way that characterized as a step. So here last step is termination. Of course this is correct. So here the wrong one is C. Now make it as C. Incorrect statement is C, not the B. Next one. Initiation factor and termination factor are initiation factor and then termination factors are initiation factor is sigma and then termination factor is rho factor respectively. So first one itself is right here. Next question, 109. Translation and transcription in eukaryotes occur in translation and transcription in eukaryotes occur in A. Cytoplasm and nucleus respectively. B. Nucleus and cytoplasm respectively. C. Cytosol and then D. Nucleus. Here the right option is B. Sorry, make it as a B. This is not correct actually. See, I will explain. What is translation and transcription in eukaryotes? See, if I am talking about this cell, inside the cell there will be nucleus, isn't it? In that there will be DNA. When transcription starts, this is a process of copying the segment, only a segment of the DNA into the RNA. DNA is copying itself in the form of mRNA, and that process is called as what? Transcription. So it takes place in nucleus. After the formation of the mRNA in the nucleus, it is going to be a maturation process, and that mRNA transferred to the cytoplasm, and in the cytoplasm with respect to the tRNA and then rRNA there will be a process called what? Translation. So where the translation happens? Translation happens in the okay. Yes, of course A is correct because respectively he is asking. Translation occurs in the cytoplasm whereas transcription occurs in the nucleus. So A itself is correct here. Okay, so don't get confused. Of course translation occurs in cytoplasm and then transcription occurs in nucleus. But when I talk about, he is asking about eukaryote, right? If I am talking about the bacteria, that is a prokaryote, here there will be a DNA, and DNA is giving or else transcribed in the form of mRNA, and with respect to mRNA, again in cytoplasm itself only translation will be occurring. So in cytosol, both the transcription and translation occurs in prokaryotes. So that's it. Not B, it's A itself is correct, because he wrote here respectively. So translation occurs in cytoplasm, transcription obviously occurs in nucleus. Next, 110. Which of the following can be coupled in bacteria? Which of the following can be coupled in bacteria? Just now I said, isn't it? I hope you people can answer. Replication and transcription? No, replication is multiplication, or else replication of the DNA, you can say. And then B, transcription and translation. Exactly, transcription and translation both occurs in cytoplasm, where in the bacteria, as I said just now. So right option is B. 111, question number 111. Transcription of 18S rRNA is done by dash in eukaryotes. Transcription of 18S rRNA is done by dash in eukaryotes, that is pol 1, pol 2, pol 3, all of this. Here the right option is A. RNA pol 1 is responsible for the rRNA formation, then processed into the 18S, 5.8S and 28S. Let me give a shortcut. See, in the eukaryotes, specifically in the eukaryotes, I said RNA polymerases are of 3 types for the transcription process. Okay, RNA pol 1 makes rRNA, pol 2 makes mRNA, and then 3 makes tRNA. Okay na? Pol 1 is responsible for the formation of rRNA, 2 is for M, and then 3 is for T. This is a shortcut trick to remember. Pol 1 is for rRNA, 2 is for M, and then 3 is for T. RMT. RMT. 1, 2, 3. RMT. Pol 1 for R, pol 2 for M, pol 3 for T. So right option is A. rRNA. Right option is A. Next, choose the incorrect statement. Choose the incorrect statement. A, 5.8 S rRNA and 5S rRNA transcribes by same RNA polymerase in eukaryotes. And then B, hnRNA and mRNA transcribed by the same RNA polymerase in eukaryotes. Here he is asking incorrect one. Okay na? First one is incorrect, because just now I said 5.8 S or RNA is transcribed by pol 1 is right, while yes 5S rRNA is transcribed by pol 3. Okay na? Pol 3. They do not use the same enzyme. They do not use the same. This is by pol 1 and this is by pol 3. That's what first statement itself is wrong. If I talk about the B, hnRNA, that is heterogeneous nuclear RNA, it's a primary transcription, and mRNA transcribed by the same RNA polymerase in eukaryotes. Of course, they produced by which one? Pol 2. And then C, tRNA and snRNA transcribed by the same polymerase in eukaryotes. Of course, that is by 3, pol 3. Okay na? So here the right option is option A, because it's a incorrect. This is only exception which is transcribed by the pol 3. Don't get confused here. So to be sure, to be clear, this is a list of polymerases. See, pol 1 is for rRNA, which makes 28S, 18S and 5.8, not the 5S. Okay, and it is found in nucleolus. Inside this cell inside the nucleus, inside the nucleolus there will be a pol 1, whereas RNA pol 2, which is a main, it is for what, messenger RNA, main messenger, and also for snRNA. This is important for the NEET, which because many times question arise from this portion for the NEET. If I talk about the pol 3, it's for tRNA and then for small RNA, that is 5s rRNA. See here it is the main. That's what pol 3 is responsible for 5s rRNA, not the pol 1. Got it? Is that clear? Along with the rRNA, pol 1 is responsible for rRNA, which is a part, that is rRNA is made up of 28S, 18S and then 5.8S. Pol 2 is for mRNA as well as snRNA, and pol 3 is for mainly for tRNA only. Along with that, it also transcribes for 5s rRNA. I hope this is clear to everybody. Next, move for the next question. 113, that is splicing is required to. Why splicing is required? It is required to remove the introns, that is interruptions. This one is correct. What happens in eukaryotes, there will be exons, introns, exons, introns, exons, introns will be there, and this one is called as hnRNA. When this is undergone for the splicing, whatever the exons are there and then introns are the interruptions, exons, introns, exons and then introns, these introns are removed and then exons are rejoined to form a mature mRNA, that is mRNA. That process is called what? Splicing, and then it is done by spliceosomes. Okay na? Exons are expressing genes and then introns are intervening genes. You can remember in interruptions manner. Okay na? Exons, introns, exons, introns, exons, introns. If the mRNA is in such manner, for the production of protein will be not there, because interruptions, inventions are there, intervenings are there. So what happens? hnRNA is going to be in the maturation process, which is called as what? Splicing. Splicing is nothing but removing of the introns and then rejoining of exons to form a mRNA, which is specifically takes place in eukaryotes. You won't see in prokaryotes. So here the right option is option A. Next question, 114. Capping is, what is actually capping is? Option A, addition of methyl guanosine triphosphate at 5' end. Yes, of course this one is correct. What actually the capping is? It's a post-transcriptional process that occurs in eukaryotes. Okay, it involves the addition of 7-methyl guanosine triphosphate. You can also write it as M7G, 7-methyl guanosine triphosphate, where at the 5' end of the precursor mRNA. Who is precursor mRNA? Pre-mRNA you can say. Why it is going to take place? First one, it protects the mRNA from degradation by the exonucleases, and it serves as a recognition signal for the ribosome to initiate the transaction. Okay, now if I say this is the mRNA, here there will be the capping at the 5' end, and at the 3' end there will be tailing also, poly A tail it is going to say. Okay, now there will be why it is called poly A tail, because adenylate residue are going to occur at the 3' end. That process is called as polyadenylation or else tailing also you can say. But at the 5' end what happens? Capping will be there. Here he is asking capping, so the right option is option A. Next, 115. The fully processed HNRNA is A. tRNA, no. B. mRNA, exactly. So the right option is option B. Heterogeneous nuclear RNA, heterogeneous nuclear HNRNA means heterogeneous nucleus RNA or else nuclear RNA. This is a primary transcript process by the polymerase II in the nucleus. It contains both exons and then introns. It contains what? Both exons and introns. That is coding and the non-coding region. After the processing, which is called as splicing, capping and then tailing. Splicing involves the removal of the introns. Capping involves addition of the methyl guanosine triphosphate cap at the 5' end, and then tailing addition of the poly A tail at 3' end. So here the right option is option B. Next question from genetic code 113. George Gamow argued A. There are only 5 bases, and if they have code for 20 amino acids, code should constitute a combination of bases. Combination of bases. And then B. There are only 4 bases, and if they have code for 20 amino acids, code should constitute a combination of bases. And C. Genetic code is triplet. Here the right option is option D, because this one is also argued by him, and then genetic code is triplet. It is also argued by him, not the B, it's D. Next question 117. Which of the following have maximum codon in genetic code? Maximum codon in genetic code, which is having leucine. Yes, of course, first one itself is right. That is, leucine has 6 codons: UUA, UUG, CUU, CUC, CUA, CUG. Okay, and whereas methionine, that is, it has only one, that is AUG, which is a start codon also. And for the cal, possibly valine, and then for the phenylalanine it has only 2 codons. So this has 2 codons and this one is having 1. So here the right option is option A. Next question, that is 118. Which of the following is or are showing dual function? Showing dual function. UUU, no. B, AUG, exactly, because in genetic code the codon AUG is a unique because it serves as initiation codon also, and it is coding for the methionine, which is a amino acid. One, it is a start codon. One more, it codes for what? Methionine. AUG is a start codon also. That's what it has a dual function. One, what it is? It acts as a starting codon. And one more, it is responsible for the, it codes for amino acids what? Methionine. So right option is here B. Next question, AUG is start and then methionine. Next question, the least number of codon is for, the least number of codon is for methionine because it's code for only AUG, whereas phenylalanine, just now I said 2, it has 2 codons. For glycine it codes for 4. Sorry, this is glutamine, it codes for 2. Glycine codes for 4. and whereas phenylalanine codes for 2. So here the right option is option A. Next, from the mutations and then genetic code. Sickle cell anemia is classical example of, already answer is revealed, that is, it's a classical example of what? Point mutation. What happens in the sickle cell anemia? What happens, single base is going to be changed. It causes by change in single nucleotide base in the DNA sequence of the gene that codes for the beta globin chain of the hemoglobin. What actually happens? A specific change is going to happen at the 6th position of the beta globin. At what? At the 6th position. Then what happens? The triplet codon, which actually should code for this is a triplet codon GAG, which codes for glutamic acid. It codes for what? Glutamic acid. Due to the point mutation, what happens? GUG will be there, okay, which codes for what? Valine. Means what? In the form, to form glutamic acid, instead of glutamic acid, it is going to form a valine. Because of that, what happens? Sickle cell anemia is going to form. That's what it is called, what? Point mutation. These cells can block blood flow and break down easily, leading to the symptoms of anemia. Next question, question number 21. In sickle cell anemia, there are changes in genes for, just now I said about that, A. Alpha globulin chain, no. B. Beta globulin chain, yes, of course. In sickle cell anemia, what happens? Caused by the point mutation in HBB gene. Where? In HBB gene, which provides instruction for the making of what? Beta globin protein, or else beta globin chain, which one you can say. Glutamic acid will be substituted by what? Valine at the 6th position of the beta globin chain. Okay, na? So here the right option is option B. Next, 122. In sickle cell anemia, resultant effect of mutation is change of amino acid residue. Change of amino acid residue, that is valine to alanine, no. Valine to glutamic acid, no. It's given opposite, isn't it? Alanine to valine, no. Glutamic acid to valine, right. So here the right option is D. Next, the following is an example of big red cap, which is read as big REM DCAP. What it is actually? What actually original sequence? BIGREDCAP. Each word represents three letters, isn't it? Similarly, how actually it is there in the DNA, it can be read in triplets. If it is mutated, like if it is mutated, what happens? See here what happens. M is added, right? M is inserted. Because of insertion of M here, overall reading pattern is changed, isn't it? That's what happens. It is said to be what? Additional mutation. What is added here? it is added M. Because of that, overall thing is shifted. The frame is shifted. That's what it is called as frame shift mutation by the addition mutation. Big red cap you can easily read, but you can't read this one because big you can, but it is added by one code, one nucleotide. That's what you can't read. Big REMDCA and then P is left because one is added here. So this is the example for what? Additional mutation. It is also called as what? Insertional mutation. Whereas deletion mutation, where one should be delete. Okay, now one or more nucleotides are deleted, removed. Point mutation, one nucleotide is swapped for another without changing the total one. Here swapping is not there, so this is not D, this is C. 124. Ram has cap. Ram has cap. 333 triplet codon is there, isn't it? Ram has big cap. The given examples show original sequence is Ram has cap. Yes, of course, 3 words or else codons I can say. New sequence is Ram has cap. Which one is added? Here has is added, isn't it? So this one is also for additional mutation only. Here total codon is added. When we discussed about here only one nucleotide is added here, but here overall codon is added. That's what it is, additional mutation. Next, about the tRNA, that is the adapter molecule. 125. tRNA has codon loop, anticodon loop, C both, and then D neither. So here tRNA has anticodon loop. tRNA is a very well known as adapter molecule because it bridges gap between the genetic code in mRNA and then amino acids of the protein. This is a anticodon loop. Actually, it's a specific region of the tRNA molecule that contains a set of 3 nucleotides called the anticodon. This anticodon is exactly complementary to the specific codon on the mRNA. What it actually means, if I say mRNA is having the codon 1, 2, 3, that specific codon, for example, if I say AUG, this is a codon, isn't it? Exactly to the complementary, there will be an anticodon which is present on the tRNA. I'm just drawing roughly. Just take it as a tRNA. Okay, here there will be a anticodon loop which will be exactly complementary. What exactly complementary to this? For A there will be U because it's RNA. As it is RNA, for A there will be U, not the T. For U there will be A. For G there will be C. This is anticodon and this one is codon which is there on the mRNA. mRNA is complementary will be there on tRNA. So this loop is called what? Anticodon loop. Next, the process of 126th question. The presence of adapter molecule to read the code on DNA and bind to amino acid was postulated by James Watson, Francis Crick, Friedrich Mischer, and then here the right option is Francis Crick. The adapter hypothesis he gave in the year 1950s. He gave the adapter hypothesis in where 1960s. Next question 127. tRNA was also called as a sRNA, that is soluble RNA. This one is right. Why it is called so? Because tRNA molecules are small and remain in supernatant. They are very small. That is, if you take the centrifugations, liquid part, they will be having the tRNA. After the larger cellular components like ribosomes and the DNA are down in the centrifuge, but the tRNA will be in the supernatant because they will be very small. That's what it is also called as what? sRNA, soluble RNA. Here the right option is A. Next 128 question. The amino acid acceptor end of tRNA is amino acid acceptor is 3-, no doubt in that. 3- is for what? Acceptor. If I say cloverleaf model or else L-shaped tertiary structure, cloverleaf shape model is secondary one. Let me draw. This is clover leaf model, and L-shaped model, if I draw, it will be somewhat like this. Okay, single standard only, not the double one. This is the L-shaped, and then this one is clover leaf. This is secondary structure and this one is tertiary structure. So here the binding site will be what? 3- end. 129. For the initiation of the translation, A. Only tRNA carries initiator amino acid to the site. No. While it is actually carries the amino acid, the term specific in the option D is more accurate, because only particular initiator tRNA can bind to the start codon at the P site of the ribosome. And then B, specific rRNA carries initiator amino acid to the site. And then C, any RNA carries initiator amino acids. No. D, specific tRNA carries initiator amino acid to the site. This one is correct. Specific, there will be a specific tRNA for specific amino acids for the initiator. So here the right one is D. Not just any tRNA can start the process. A specific initiator tRNA can only recognize the start codon, that is AUG, as I said on the mRNA. So this one is correct for the initiation translation. Next is 130 for the stop codon. For the stop codon, there are specific tRNA with amino acids. No. There are specific tRNAs which do not binds to the any amino acids. No, there is no tRNA. This one is correct. As like the initiation codons or else initiation tRNA, there is no such kind of codons for the anticodons for the stop codon. Okay, so the right option is option C. The given figure shows what? The given figure shows secondary structure of tRNA, that is clover leaf model, primary structure of tRNA clover leaf, secondary structure of the tRNA inverted L, and then primary structure of the tRNA, that is inverted L. So this is the secondary structure of tRNA, that is clover leaf model. Next, translation. 132th question. Translation refers to the process of, already answer is revealed, polymerization of amino acids to form a polypeptide. That is called what? Translation. Next, 133. The order and then sequence of amino acid during translation are defined by A. The sequence of bases in a rRNA? No. The sequence of bases in tRNA? No. The sequence of bases in mRNA? Right. So here the right option is C. Whatever the amino acid, the order of sequence. See, if I say this is the amino acid chain, this sequence of amino acids chain during translation are defined by whom? Because whatever the codon is there on the mRNA, that is 3 codon will give 1 amino acid, and 1 more 3 is responsible for this. So whatever the sequence of bases on the mRNA is there, that is responsible for the order of sequence on the amino acid. That's what this is, right. 134. Which of the following bond is formed during translation? I said many. I said use many of the time this word. Glycosidic bond? No. Phosphodiester bond? No. Peptide bond? Yes. So here the right option is C. Glycosidic bond, these are the bonds linked between the sugar molecules together to form a carbohydrates. If I talk about the phosphodiester bond, I explained about this, isn't it? These are the bond which is linked between the 2 nucleotides together to form a backbone of the DNA. So here the right option is peptide bond, which is responsible to form amino acids. Next, 135. First phase of translation does not involve charging of RNA. Amino acids are activated in presence of ATP. Activated amino acids are linked to their cognate tRNA. None of this. Here the right option is none of this. Because charging of an RNA, amino acids are activated, these all processes are going to take place. First phase, all are essential for the first phase of the translation. That's what. Next, 136. The initiation or the first phase of translation is A. Amino acylation of tRNA. Exactly, this answer itself is right. Also, it is also called what? tRNA charging is essential first step before the translation can be begun at the ribosome. Amino acids must be activated and linked to their specific tRNA, that is tRNA molecule. This reaction itself is called what? Amino acylation. Next question, 137. The cellular factory responsible for the synthesizing protein is ribosome, lysosome, peroxisome, none of this. Here the cellular factory is called what? Ribosome. Next, 138. In inactive state, protein factory of cell exists in inactive state, protein factory of the cell is exist in 2 states, 4 states in prokaryotes, 6 states in eukaryotes, B and then C both. So here the right option is A. What actually this one is mean? In inactive state, the protein factory, what is protein factory? Ribosome. In cell, where it is, see, if I take this as a cell, there is no translation is going on Okay, before that what happens, the larger and then smaller subunits are not attached to one another. That state itself is called what? Inactive state. So protein factor, it is ribosome. If they find out mRNA, then what happens, larger subunit will be there and then smaller subunit is going to attached. So this one is active stage and this one is inactive state. Okay now? Yes. Next, 139. Which of the following is sign as beginning of translation? Sign as a beginning of the translation. Here, when the large subunit of protein factory of a cell encounters an mRNA. B. When a small subunit of protein factory of the cell encounters an mRNA. C. When a small subunit of protein factory of cell encounters a tRNA. D. When the larger subunit of protein factory cell encounters the tRNA. So here the right option is option B, because whenever the mRNA is come from the nucleus, what happens, the smaller subunit will be encountered, then next what happens, there will be the encountering of the larger subunit. So here the right option is option B. So the next question is, that is question number 140. The bond formation, that is peptide, between the charged tRNA is accomplished due to option A. Presence of ATP and catalyst. Yes, this is correct. Option B. Two such charged tRNA are brought close by two sites in the large subunit of the ribosome. Yes, of course, this is also correct. What actually happens? The formation of peptide bond is an energy consuming process. Okay, now the energy is derived from the ATP. That's what this is correct. And which is used during the charging of the tRNA, that is amino acylation, just now I said, isn't it? Additionally, the ribosome itself acts as a catalyst, specifically the ribozyme peptidyl transferase. Why the B is correct? Two such charged tRNA are brought close by two sites in the large subunit of the ribosome. Yes, of course. For a peptide bond to form, the two amino acids must be physically close to each other, right? So this happens in the large subunit of a ribosome, where A site and then P site bring the two charged tRNA into close proximity. Okay, now if I show you tRNA, sorry, this is rRNA. Sorry, this must be a small, and there will be mRNA. Here rRNA large subunit has E site, P site and then A site. What is A? Acceptance of the tRNA. P is peptide bond formation site, and then E, exit site. Exit site for what? Exit site for polypeptide chain, also for the tRNA also. So here what happens among these two? If I say already polypeptide chain is forming, to this polypeptide chain one more polypeptide chain is adding, means the specific tRNA is going to attach here, and that is going to be near to them, okay, to form a bond, like that it will be. So here the right option is option D. Both A and B are correct. Next, 141. The ribosome acts as a catalyst during the bond formation, as in A. 28S rRNA in bacteria, and then B. 23S rRNA in bacteria. C. 23S rRNA in eukaryotes. D. 28S tRNA in bacteria. So here the right option is option B. In bacteria, the ribosome acts as a catalyst for the formation of peptide bonds during the protein synthesis, specifically ribozyme activity, that is 23S rRNA. Next question, that is question number 142. Choose the correct statement. Option A. A translation unit in mRNA is sequence of RNA that is flanked by start codon and stop codons and codes for polypeptide. This one is correct. This statement itself is correct, means no need to read the other one. So yes, option A itself is correct. Next question, that is question number 143. UTR is or are A. untranslated region of mRNA, correct. 2. It presents at both 5' that is start codon and 3' before the stop codon. Yes, of course, UTR's 5' is located upstream of the UTR. For example, if I say this is the mRNA, UTR is located upstream of the start codon, that is AUG, and UTR is located downstream of the stop codon also, that is UAA, UAG or UGA. So here the statement 2 is also right. Next, third one, they are required for the efficient translation process. Yes, this is also correct. For the efficient translation process, they are needed. And then for the fourth, it present at both 3' before the start codon and then 3' after this codon. No, this one is wrong. So 1, 2, 3 are correct, and then fourth one is wrong. 1, 2, 3 are correct. So here the right option is option A, not option D. Option A is correct. First one is correct, and then second one is correct, and then third one is also correct. They are required for efficient translation. Yes, of course it is correct. So not the D, make it as A. Next question, 144. Initiator tRNA binds with. Initiator tRNA binds with AUG codon of mRNA. B. At initiation of the protein synthesis. Yes. ATG codon of double-stranded RNA. No, this one is wrong. So both A and B are correct. So the right option is D, A and B both. 145. Choose the correct about elongation of translation. Elongation of translation. Choose the correct one. Here A. Complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. This is correct. What happens during the elongation? Charged tRNA move into the ribosome, and then tRNA anticodon recognizes and binds to the matching mRNA codon. B. The ribosome moves from codon to codon along the mRNA in 3' to 5' end. Up to this is correct, but not the 3' to 5' end. It's from 5' to 3'. Reading is from 5' to 3'. So this one is wrong. Correct, he is asking, isn't it? Next. Complexes composed of an amino acid linked to tRNA sequentially bind to appropriate anticodon in mRNA by forming complementary base pairs with the tRNA. The mRNA contains codon and the tRNA contains anticodon, not the other way around. Sequences bind to the appropriate anticodon. Here they are given anticodon in mRNA. They given anticodon will be there in tRNA, not codon. Codon will be there on mRNA. So this one is wrong. Choose the correct about the elongation translation. So here the right option is option A. Only this one is right. Next question, that is question number 146. Termination of translation complex is done when release factor binds with the stop codon. No. Release factor binds with the UGA like codons. Correct. So here the right option is option B. Next, 147 from the regulation of gene expression. Gene regulation in eukaryotes exerted at A. Formation of primary transcription, B. Transport of mRNA from nucleus to the cytoplasm, C. Both A and B, and then D. Regulation of splicing of the tRNA. So here the right option is option C, that is A and B both are correct. Next, 148. Beta galactosidase is used to catalyze the hydrolysis of it must be A. Lactose into galactose and then glucose. Yes, of course, the function of beta galactosidase is lactose into galactose and then glucose. It hydrolyzes. Next question, so here the right option is A. Next question, that is question number 149. E. coli do not have lactose around them to utilize for the energy source. They would A. no longer require the synthesis of enzyme beta galactosidase. No. Synthesize enzyme beta galactosidase. C. die due to the lack of carbon source and then energy source. This one is also wrong. So none of these are correct with respect to that. Next question, 150. In prokaryotes, predominant site for control of gene expression is predominant site for the control of gene expression is A. control of rate of transcriptional initiation, B. control of rate of translation, C. control of rate of transcriptional elongation, D. both B and C. So here the right option is A. control of rate of transcriptional initiation. It's predominant. Next question, that is question number 151. Given below our statement, choose the incorrect statement. A. the development and differentiation of embryo into adult organisms are result of coordinate regulation of expression of several sets of genes. Yes, of course, this one is right, because when the embryo is going to adult, that is embryo development is a highly complex and then tightly regulated process where different types of the sets of the genes are going to turn on and then off in a specific sequences to form a various tissues and then organs. So this one is right. B. regulatory proteins act positively in activator. Yes, of course, they are going to act as a positively also and then they act also negative also. C. in transcriptional unit, the activity of RNA polymerase at a given promoter is in turn regulated by the interaction with the accessory proteins. Yes, this one is also right. So there is no incorrect statement. So the right option is option D. Next one, next question, that is question number 152. Operator. The region adjacent to sequence by which repressor mRNA formed. B. bind with repressor protein. C. bind with inducer, and then D. both A and B. So here the right option is option B, bind with repressor protein. What actually the operator? With respect to the lactose, lac operon, that is lac operon, we have P, I, I gene, P gene, I gene, and one more P, and here there will be operator and there will be structural genes, isn't it? Z, Y, A. Whatever the I gene is going to form the mRNA, that mRNA is forming repressor. This is a repressor. If lactose is there, that lactose is going to bind the repressor, and then repressor is not going to bind to the operator. Okay, so here bind with the repressor protein means if lactose is absent, this is going to bind here. So that's what this one is right. If I say A1 is also right, this is a region adjacent to the sequence by which repressor mRNA is formed. This mRNA is forming, no, this is adjacent to the sequence of which repressor mRNA is formed, right? It is adjacent only, but we'll go with this exactly answer. Okay, next 153. Each operon has option A, same operator and same repressor. Option B, same operator but specific repressor. Option C, specific operator but same repressor. Option D, specific operator and specific repressor. So here the right option is option D. It has a specific operator and a specific repressor. Next, with respect to the lac operon concept, this concept is very important for your NEET point of view because much more questions you can see. 154. Lac operon was studied first by Francois Jacob, Jackie Monod, geneticist and biochemist. D, none of this. Here the right option is option C, geneticist and biochemist. 155. Lac operon is A, monocistronic structure gene, is regulated by a common promoter and then regulator. No. Why? Because it is not mono, it's a poly. B, polycistronic structural gene is regulated by a common promoter and a regulator gene. This one is correct. So option B is right. Next, 156. The regulatory gene of lac operon is P gene. No. I gene, yes. O gene, no. And then Z gene, no. Here I gene is responsible for production of repressor protein, that is the gene which is responsible for the formation of the repressor protein. Here it is what I means, it a inhibitor. It means I stands for what? Inhibitor. It is responsible for the switch off and then switch on. It ensures that operon is switched off when the lactose is not present. If lactose is present, it will be switched on. That process we are going to discuss. What is P gene actually? It's a promoter. What is O gene? It's a operator. And then Z is a structural gene which is responsible for the which codes for the beta galactosidase. So here the right option is option B. 157. I in I gene stands for, just now I said, isn't it? Inducer, no. Inhibitor, yes. So here the right option is B, inhibitor. Next question, 158. Match the following one with following two. Column 1 with column 2. Gene Z, A, Y and then I are given. If you know any two, you can guess the right option very quickly. Z gene, which is responsible for the production of beta galactosidase, that is beta gal, we can also say. Z is 2. A is 2 means A is 2, the last one. Let me cross check. A gene is responsible for the transacetylase. B is 4. B is 4. So right option here it is D. So quickly you can remember by this. Proper regulation of ZY action. Keep in mind this statement. Proper regulation of ZY action. Here P is PROZYA. P is for promoter, R is for regulatory gene, and then O is for operator. What is regulatory gene? That is I, we can indicate. And then Z, which produce beta galactosidase, and then Y produce permease, and then A, which is code for transacetylase. So PROZYA. Just keep in mind this statement. Proper regulation of ZY action. Next question, that is question number 159. The monomeric product of lactose is chiefly hydrolyzed by. Monomeric product of lactose is chiefly hydrolyzed by A gene, Z gene, A gene, and then Y gene. I gene, Z gene, A gene, and Y gene. Here the right option is Z gene, because Z gene codes for beta gal, and the primary enzyme that hydrolyzes the lactose into monomeric units, that is glucose and galactosidase. It is responsible for the beta gal, and beta gal is going to hydrolyze, that is break down the lactose into what? Glucose and galactose. So here the right option is option B. Next question, that is question number 160. Lac in Operon is for A, monosaccharide, B, disaccharide, C, polysaccharide, D, insect. Here the right option is option B, disaccharide. And the next question, question number 161. In absence of preferred carbon source, if lactose is provided in growth medium of the bacteria, the lactose is transported into cell through the action of product formed by. Here option A is I gene, B Z gene, C A gene, D Y gene. And here the right option is option D, Y gene. Y gene codes for enzyme permease. This enzyme is responsible for increased permeability of the cell. For what? For beta galactosidase, effectively transporting lactose into the cell. So that's what it is responsible for, the permeability, which is releasing the enzyme permease. 62. Allolactose is, next question, that is question number 162, sorry. Allolactose is A, inducer of lac operon, B, inductive repressor, C, form of lactose that bind with the product of repressor mRNA and inhibits transcription of the structural genes. D, all of this. Here the right option is option A. It's an inducer of lac operon. It's isomer of lactose actually. When lactose enters the cell, a small amount of converted into the allolactose by the enzyme beta galactosidase. Next, 163. Lac operon is A, negative regulation operon, B, positive regulation operon, C, A and B both, D, none of this. Here the right option is C, A and B both. Next question, 164. Inducer of lac operon is inducer of lac operon is A. Glucose, B. galactose, C. lactose, and then D. fructose. Here the right option is C. lactose. Next question. Here in the given diagram, A, B, C, D, E, F, G is given. What it is indicating? The given diagram is in presence of lactose. The given diagram is in the absence of lactose. The given diagram is of a gene of D and H is the same process. So here the right option is option A. Why it is A? Because see here, from the I gene mRNA is produced, and from that mRNA repressor is produced. When this repressor is there, the lactose is going to combined with the repressor and then acting with the repressor, so that what happens, this repressor, inactive repressor, what it is not at all able to join or else attach to the operator gene, so that what happens, the transcription happens. So mRNA is produced by the Z, Y and then A gene, which is responsible for the formation of beta gal, permease and then transacetylase, so that in the presence of what, inducer happens. So this is the in the presence of lactose. Here inducer who, lactose. Lactose present means switch on condition. If the lactose is absent means switch off condition. Next, from the questions, human genome project. Here from this concept I can bet that you will get compulsorily one question. Sure, damn surely you will get one question from HGP. That much important concept this one is. So 166, HGP was launched in 1980, 1970, 1990, 2000. It is launched in 1990. HGP was called a major project, hexagonal project, mega project, and none of this. It is called as what? Mega project. Why it is called as mega project? Because of many reasons. Because its scale and then scope, it was a massive international scientific research project with a goal of determining the base pairs that make up the human DNA. Means whatever the human DNA is there, no, approx 2 meter of the human DNA, whatever the base pairing is there, no, that sequencing project it was. And even though it is called mega project because of the cost, because $3 billion, roughly $1 per base pair, that much cost it is. And data magnitude and then time and then resource, the project spanned 13 years and required the collaboration of the numerous scientists, high speed computational devices and advanced technologies worldwide. That's what it is called, what? Mega project. Due to the what? Due to the time and then resource, because it took 13 years, due to the cost also. Many reasons are there. Next question, 168. Human genome has approximately dash base pair. Human genome has approximately dash base pair. A, 3 into 10 to the power 9, 3 into 10 to the power 6, 6 into 10 to the power 9, 6 into 10 to the power 6. It must be power, but it's given like that. Here the right option is option A. Next question. Here is a trick. 3 into 2, 99. How you can remember 3 to 99? 3 billion base pairs are there. 20 to 25,000 genes are there in the human genome. 99% same in the humans. So 3 billion, 20 to 25,000 genes and 99% same in the humans. Only 1% we differ from one another. Almost all base pair is similar in me and you also. Got it? Next. Here, next question, that is 169. If cost of sequencing required is $3 per base pair, $3 per base pair, then total cost of sequencing human genome as per 8 to 3 will be A. US dollars 18 billion, B. US dollars 9 billion, C. US dollars 18 million, D. US dollars 9 million. Here simple super trick is 3 is given, 3 into 3, 3 base pairs are there, so 3 into 3 equals to 9. Just add billions here, so that option here it is option B is right. But calculative wise, if you ask, here is the calculation how you can calculate. What they are given, cost per base pair is 3, cost per base pair is 3, and human genome size as per given in the NCERT 3 into 10 to the power of 9 base pair, isn't it? If you multiply 3 into 3, the given base pair cost and then human genome size, then you will get 9, and then this 10 to the power 9 base pair is adding there. So final answer is 9 billion. Formula, total cost equals to cost per base pair and then how much base pair it is having. Now next, goals of HGP. One 70th question. There are dash genes in humans DNA as per the goal of the HGP. Just now I said 20 to 25,000. So here the right option is option A. Next, 171. HGP was a dash year project. How many years? 13 years. So answer C is correct. 172. HGP was coordination by A. US Department of Engineering and National Institute of Health, B. US Department of Engineering and National Institute of Biotechnology, C. US Department of Energy and National Institute of Biotechnology, D. US Department Energy and National Institute of Health. So here the right option is option D. Next question, 173. The dash of UK was the major partner of the HGP. Dash of UK was the major partner of the HGP, that is A. Welcome Trust, B. Health Trust, Social Trust, and then D. Welcome Trust. Here the right option is option A. Both the options are same, but the spelling is different, isn't it? Next, 174. The project was completed in A. 2005, B. 2004, C. 2003, D. 2002. Here the right option is 2003, C. It is launched in the year 1990 and then it is completed in the year 2003. Next question, 175. Additional contribute to the HGP was A. Japan, B. China, C. Germany, all of this. Here the right option is all of this. The human project, that is HGP, Human Genome Project, was an international scientific research project. While it was primarily coordinated by the US Department of Energy and the National Institute of Health, we discussed about that. Several other countries made significant contributes to the mega project. According to the this major partner is UK, that is United Kingdom, Welcome Trust. Additional to that, Japan, France, Germany and China. Next question, that is 176. Senor habitis elegans is A. fungi, B. nematode, C. bacteria, D. virus. It's a nematode. Next question, that is 177. Senor habitis elegans is a free living non-pathogenic, B. a pathogenic parasitic, C. free living pathogenic, D. parasitic non-pathogenic. Here the right option is option A, free living non-pathogenic. 178. Methods and approaches of HGP includes. Methods and approaches of HGP includes A, excess sequence tags, B, expressed sequence tags, C, excessive sequence tags, and then D, exerted sequence tag. Here the right option is option B, expressed sequence tags. Next question, question number 179. Sequence annotation refers to A, identifying all genes expressed as RNA and then sequencing them. B, sequencing the whole set of genome and then assigning different regions with function. C, identifying the sequence the genome simultaneously. D, more than one option is correct. And then here the right option is option B, sequencing the whole set of genome and then assigning different regions with the functions. Next, 180. For sequencing the DNA is A, partially extracted from cell, totally isolated from cell, not needed to isolate from cell, none of this. Here the right option is B, totally isolated from the cell. Next question, 181. The DNA for sequencing is converted to fragments of small size. The fragments are made A, on a pre-decided basis, B, on a predefined basis, C, randomly, D, depend upon the organism. Here C, randomly. In the modern DNA sequencing, what happens, a shotgun sequencing method is there, which is a standard reference in biology like NCRT. The total DNA of an organism is first isolated and then broken into smaller fragments. And then what actually the random fragmentation means, this fragmentation is done randomly using methods like sonication or a specific enzyme. Okay. Next, 182. The step in DNA sequencing after fragmentation of the DNA is. The steps in the DNA sequencing after fragmentation of DNA is. First one, cloning in host using vector. B, cloning in vector using host. C, amplification of DNA fragment. And then D, more than one option is right, because option B and then option C. 183. Commonly used host for DNA cloning includes A, bacteria, B, BAC, C, Yak, and then D, both A and C. Here the right option is both A and C. Yak stands for yeast artificial chromosome and then BAC stands for bacterial artificial chromosome. Yeast artificial chromosome, bacterial artificial chromosome. Next question, 184. Just now I said about this, isn't it? BAC stands for bacterial artificial color, no. Bacterial artificial characterization, no. Bacterial artificial chromosome, right. So here the right option is C. Next, fragments were sequenced using automated DNA sequence that worked on principle of method developed by Erwin Chargaff, Marshall Nirenberg, Frederick Sanger, and then George Gamow. Here the right option is C, Frederick Sanger. That sequence itself is called as Sanger's method or Sanger's sequencing. Next question, 186. Method for determination of amines acid sequence in proteins were developed by Erwin Chargaff, Marshall Nirenberg, Frederick Sanger, George Gamow. Here the right option is C, Frederick Sanger. Next, 187. The last of the 24 human chromosome to be sequenced was. The last of the human chromosome to be sequenced was A, chromosome 1, B, chromosome X, C chromosome 22, D chromosome Y. Here the right option is option A, chromosome 1. Although chromosome 22 was the first human chromosome to be fully sequenced, chromosome 1 was the last of the 24 human chromosomes to have its sequence completed and published. Next question, silent features of human genome concepts. 188. According to HGP, human genome contains, according to HGP, human genome contains A, 3000 million base pair, B, 6000 million base pair, C, 9000 million base pair, and then D, million base pair. So here the right option is 3. Next question, 189. Dystrophin was found to be A, largest known human gene with 2.4 million bases. Yes, of course, this one is correct. So here the right option is option A. 190. Which chromosome was found to have most genes? Which chromosome is found to have most genes? Here option A, chromosome 22, chromosome 1, 5, and then Y. Here the right option is chromosome 1. As the largest human chromosome, it contains the highest number of genes. Which one? Chromosome 1. It is estimated to have approximately 2968 genes. Which one? In the chromosome 1. How much? 2968. And then Y chromosome is the one of the smallest one, which is having 231 genes. So the next question, that is 191. Which chromosome was found to have a fewest genes? Which chromosome was found to have a fewest genes? A, chromosome X, chromosome Y, C, chromosome 1, D, chromosome 5. Here the right option is option B, chromosome Y. Next one, from the DNA fingerprinting. 192 question. The DNA sequence in which small stretch of DNA is repeated many times is called A, SNP, B, repetitive DNA, C, polymorphic DNA, D, more than one option is right. Here the answer is option B, repetitive DNA. Next, 193. Satellite DNA classified into different categories like microsatellite, mini satellite, and etc. based on what? Based on A, length of segment, B, number of repetitive, C, base composition, D, all of this. Here the right option is all of this. 194. Polymorphism arises due to A, mutation, inheritable. So this one is right. 195. DNA polymorphism is observed more in A, non-coding DNA sequence as its mutation affects reproduction, B, coding DNA sequence as its mutation affects reproduction, C, non-coding DNA sequence as its mutation may not affect the reproductive ability. So this one is the right. Here the right option is option C. 196. Technique of DNA fingerprinting was initially developed by James Watson, B, Jansley, C, Alec Jeffrey, D, Maheshwar. So here the right option is Alec Jeffrey. 197. VNTR stands for, already option is revealed, that is D, variable number of tandem repeats. VNTR belongs to microsatellite, macrosatellite, mini satellite, all of these. Here the right option is C, mini satellite. VNTR is a part of mini satellite. 199. In the given figure, if C is DNA collected from the crime site, A and B are samples from the suspects. Then who is the criminal? If C is DNA collected from the crime site, A and B are the samples of suspect. If A and B is suspect, who is exactly similar? B is similar, isn't it? C. So B is correct. Answer is A. Last but not the least, question number 200. Which of the following forms the basis of DNA fingerprinting? A. Differences in number of genes. B. Variations in VNTR sequence. C. Protein sequence variation. D. RNA expression levels. Here the right option is B. Variation in the VNTR sequence. So this is the trick to remember what actually is going on in the DNA fingerprinting. CEPTAV, CEPTAV. First, collection of sample, extraction of DNA will be there. Then next, PCR amplification, polymerase chain reaction amplification will be there. Then next, cutting from the enzyme, restriction enzyme. And then next, gel electrophoresis. Next, visualization. So how come you can easily remember this one? Collect, extract, prepare, test, analyze, and then view. So this is the steps which will be involved in the DNA fingerprinting. I hope everybody is understood whatever we discussed today. And one of the most interesting chapter, as I said, this is having more marks weightage also, and then interesting also. And with the all of the tricks and then shortcuts whichever I have given, just please keep in mind and then practice more, because the exam is near. From the DNA double strand helix to protein formation, you have just unlocked the core secret of life, isn't it? Remember, every trait, every function in your body starts from this molecular code. If you master this chapter, you are already ahead in the NEET biology. Revise smart, practice constantly, and don't forget, concepts make topper, not just the memorization. If this helped you, like, share, and then subscribe for my channel, that is BioPulse. Let's crack NEET together. Thank you, one and all.